Well, good morning or good afternoon, wherever you're calling in from, and welcome. Today we are discussing research in the cognitive psychology domain using wearable eye tracking. This Researcher Spotlight series is a collaboration between Tobii and our customers to highlight groundbreaking research utilizing our eye tracking systems and meant to showcase current and ongoing work to hopefully give you inspiration, answer questions, and create networking opportunities. This webinar will be recorded, so feel free to rewatch or share the link with students and colleagues. A link to this webinar recording will be sent by email to everyone who registered within a few days. We've also hosted other webinars like this if you're interested, and they can be found in our Tobii Resource Center. Here is our agenda for the day. I am so honored to host a group of researchers from the US and Europe. Each presentation today will be approximately 15 minutes, and after all the presenters have finished, we will begin the roundtable discussion where you can ask questions directly at the presenters. For this webinar, each researcher has used our wearable eye tracking systems, so I want to show you briefly how they work. Our Tobii wearable eye tracking hardware consists of multiple components, including cameras, near-infrared illuminators, and algorithms. The cameras take pictures of the eyes at either 50 or 100 hertz, so that equates to about every 10 or 20 milliseconds. The illuminators create a corneal reflection on the eye, which then allows the algorithms to determine where the participant is visually attending. There's a scene camera in the front to capture where the head's oriented, and then on the recording you can see the 3D gaze point overlaid on that scene camera recording. The unit is powered by a lithium-ion battery in the recording unit, which can be comfortably worn for mobile research. The data is then stored on an SD card, and it can also be streamed live to watch through the eyes of your participants in real time. You can also analyze qualitative and quantitative data offline. Today, our researchers are presenting data collected with either our older model Tobii Pro Glasses 2 or our newest wearable Tobii Pro Glasses 3. Both of these hardware are compatible with Tobii Pro Lab and our Tobii Pro Glasses API. But our portfolio contains more than just the wearable eye trackers. We also have screen-based eye trackers with sampling rates up to 1200 hertz. And then these systems are also compatible with the same Tobii Pro Lab software, as well as our Tobii Pro SDK. We even offer solutions together with our partners, so if you're interested in synchronizing eye tracking data with other types of physiological data streams like EEG, GSR, heart rate, you can do so. Tobii Pro Lab is our software platform for design, recording, data inspection, and analysis. Some of the design features include an easy drag and drop interface for images, videos, text, web, or screen recording. We also have advanced options for sampling and randomization with groups and hierarchies. We have open-ended participant variables for data management and easy co-registrations with TTL sync out included at no extra cost. It's just a little toggle button. Some of the record features, we can capture participant gaze, their participant camera and audio. You can even capture GSR data with the Shimmer Plus device right here in Tobii Pro Lab for full eye tracking and GSR integration. And then for the analysis features, you can replay video recordings, flexibly draw AOIs, create custom times of interest, view and export qualitative visualizations like gaze plots and heat maps. And then there's a variety of Excel compatible metrics for full data export in TSV format. This is a webinar specifically highlighting research in the broad field of cognitive psychology, but eye tracking can really be used in almost any field where you're interested in studying human attention. Eye tracking provides a rich data set from specific metrics like fixations or saccades to eye-related biometrics like pupil diameter, eye openness, and blinks. These provide unique insights into the cognitive state of humans from attention and memory to decision-making and problem-solving. So now we're going to learn more about some unique findings in the world of cognitive psychology research. I'll give you an overview of our presenters today. We have Alana Muller from the University of Arizona in the United States, Dr. Alexandra Hoffman from the University of Innsbruck in Austria, and finally, Dr. Daniel Levin from Vanderbilt University here in the US as well. So with that, we'll get started. I'd first like to introduce Alana Muller from the University of Arizona. She's going to talk about her work in comparing the impact of vision and body-based cues to memorize spatial layouts. And now over to you, Alana.
Okay, I think I'm unmuted now. Thank you for that introduction. I just want to make sure, okay, I think everyone can see my screen. So my name is Alana Muller. I'm a fourth-year graduate student at the University of Arizona, and I use the Tobii Glasses 2 to study eye tracking in a real-world environment in addition to memory. And so I'll be telling you about my project, comparing the impact of vision and body-based cues to memorize spatial layouts. So humans are inherently visual creatures. Usually when vision is available, and especially in environments we don't know yet, we walk into a room, look around, and decide on a goal, and then move our bodies there using our body-based cues. And so we've been studying this for a long time, and yet the specific contributions to each have not been fully fleshed out yet. It's still unclear. One of the best ways you might think to study this is with the most naturalistic visual cues and body-based cues. And so that would be something like an external environment, but unfortunately, these are very difficult to control. So this is a picture of downtown Tucson, the city where my university is located. And if I wanted to study how people get around in cities, then I might try to put someone in downtown Tucson and have them walk around. But there are a lot of things about the environment you can't control. The traffic, the pedestrians, people trying to give you flyers on the sidewalk, but also things about the participant themselves, like their knowledge of the area. If I had someone who was born and raised in Tucson, they should know the area better than somebody who just moved there for undergraduate. So one of the ways that you could account for this is using virtual reality. Yet that introduces a couple of other limitations. So with virtual reality, you're often looking at a 2D screen like the picture on the left, and this doesn't give you the most realistic visual cues that you could have. You could also have an HMD that will head-mounted display on the right side of the picture that will let you walk around in an environment, but you're still faced with those impoverished visual cues. You could also have something like this device on the right called an omnidirectional treadmill, in which you can have very large virtual environments and walk around in them using this treadmill. And it's a low friction surface on the bottom, so you can kind of slip on the surface and simulate walking. And yet these body-based cues feel very unnatural when you're actually walking in the environment. So VR can account for some of the limitations that external environments present, but then they have some of their own limitations. In addition, sometimes objects that are not real are different in behavior and neural recordings. Some studies have found that real objects elicit different behavior and different memory and recordings than just pictures of objects, and that 3D objects are often more memorable. So if you look at something like this line drawing of a volleyball versus a cartoon drawing of a volleyball versus an actual picture of one, these would elicit different neural response and behavior to actually holding a volleyball in your hands. And sometimes a difference like this might not be very important, but sometimes it can be very important. So it is important that we have studies that do both, that do very simple stimuli and have very controlled experiments, and some that do broader work that maybe encompass a lot and have a lot of what we call ecological validity, which is a term that describes studying behaviors that are actually seen in the real world. And that was one of our goals of this study. So the reason we chose to go with eye tracking glasses was because we saw this as a solution to bridge the gap between hard to control environments and some of the limitations of VR. If you have eye tracking paired with natural free ambulation in an environment, then that should give you the best of both worlds, really. So you have naturalistic views and you're walking with your own body. And that will allow us to see where your eyes are actually going to. The eye tracking is really important to studying navigation with vision, because it's not enough to just know that vision's available. We need to know what people are specifically looking at and for how long, and that will help us understand how people are navigating in real-world environments. And so this work has already started to become represented in the literature. There is an example on the right here of an older adult walking around in a real-world space and someone walking around outside. So this work is starting to pick up traction, but it's only in the past couple of decades that we've seen technology that can allow us to adapt to some of these problems that eye trackers have historically had, such as having to be chin fixed to kind of a post and needing to keep your body really still to study how your eyes are moving. And we know that eye movements are tied to some cognitive processes like memory, which makes it even more important to track in our study. So there are examples of mobile eye tracking that have been used in pedestrian navigation and memory for learned routes. And so we'd like to add to the literature about this free ecologically valid designs where people are walking around in a real world environment with these mobile eye tracking glasses. And so we're after a very highly ecologically valid study using very real stimuli and having people walk around in a room themselves. So with that, our research questions involved how much participants could learn just using their eyes and how eye movements could tell us about memory retrieval, search accuracy, and navigation. We're also interested, let's see, slides aren't catching up. There we go. We're also interested in how much additional information body-based cues could give us in addition to vision. And finally, we wanted to know, while people were in this small environment we have the experiment set in, how do landmarks in the room play a role in memory? For our equipment, we use the Tobii eye tracking glasses, the Pro Glasses 2. We also had a face shield that participants would use, and we could instruct them to block their vision at certain times that we didn't want them to see. And they also had a mask to mitigate COVID. For our stimuli and environment, we put people into this 6 meter by 6 meter room. And you can see on the left and right walls that there are objects attached to it. We had these very lightweight objects that had Velcro on the back of them that could attach to the walls of excuse me, of our room. And participants would have to memorize the locations of these items in the room. There were 4 items on each wall per trial, and each participant saw 10 trials. And a trial consisted of one room with 8 objects that they would memorize, take down, and then put back up, and the trial would reset. So the procedure was a study phase and an object removal phase, and then finally a test phase to assess their memory. For the study phase, participants stood in one specific spot in the room, and they memorized the object locations for 30 seconds, and they were told that they would be tested. So they memorized with the expectation that they would have a test. Then in the object removal phase, this happened with no vision, so we asked them to put the visor down and block their vision, and it happened in one of 2 conditions. Either the experimenter alone would walk around the room and collect objects in the portrayed volleyball cart, and in the other condition, the participant would go with the experimenter, still blocked vision with the visor down, and the experimenter would let them know when something was on their left or right, so the participant could actually reach for it and take it down. Then the test phase happened in one of 2 conditions. In the first condition, the participant would be put back in the same spot that they encoded, and they would be asked to put everything back where they remembered it being. And they would use the volleyball cart and actually walk around the room themselves to put objects back onto the wall. In the other condition, they were sent to the other side of the room and instructed to put the objects back starting from that location, but they could still walk around the room as long as they didn't walk back to the beginning location to check their work. In this condition, there was no time limit, so they could take as long as they wanted to. Our hypotheses were that body-based cues would help in the event of going to the other side of the room, because we thought that beginning retrieval at the same place as encoding would be easier. So having to start from the opposite side should lead to less accuracy, but having extra body-based cues to help them remember where things were may help boost that accuracy. We also thought that having more fixations and longer durations of fixations would lead to better placement, and so lower placement error. We also thought that memory would be better when objects were located next to local wall landmarks. So for our first hypothesis, the way we got the information for placement accuracy was we had several cameras set up in the room. You can see them pictured very small in these pictures I have. These videos were then separated into pictures of the wall at study and then after retrieval, and went through a couple of video processing alterations to fix the fisheye lens and the perspective. And we ended up with two almost identical looking photos, except that you could tell objects had been moved. So we figured out the hypotenuse of where these objects were moved using the pixel coordinates, and then we transferred or converted pixel coordinates into centimeters. And so that is the measure that you'll see on the y-axis for placement error. It was also very skewed, so we log transformed it, which is why the label is log centimeters. And this is the main result we found for our first hypothesis. You can see that this looks very much like a flat line, and we did not find an effect of walking or viewpoint on our data, which was very interesting for us. But this does imply that vision was sufficient to memorize everything from one point of view for 30 seconds during encoding, and that having additional body-based cues during the object removal phase didn't help boost accuracy in any condition. So for our second hypothesis, we used the Tobii Pro Lab software, and it was really helpful to use the assisted mapping feature. That feature would take a 2D picture that you upload and plot the gaze from the video onto that picture. And it was very helpful to have that and then do a manual check yourself to make sure the gaze points were being plotted correctly. And you could also manually fix those. So I thought that was really good for the software. It was very helpful. We also used the areas of interest tool to draw around each object and each landmark. So this did take a while, and having so much data for my project did slow down the program a little bit, but it was still incredibly helpful. And so that gave us this plot here, which you can see on the x-axis, average duration of fixations in milliseconds, and placement error again on the y-axis. And we did find a significant correlation for average duration of fixations and placement error, in that the longer you looked at an object, the more that you placed it correctly, the lower the placement error was. However, we didn't find this for number of fixations, which I didn't include for time purposes. Our third hypothesis was that memory would be better for objects located on local wall landmarks, which we decided, you can see in this picture, everything in red we decided was a local landmark, something on the wall that could not be removed or covered in our experiment. So objects that were near these landmarks were actually more memorable than objects that were not directly next to a landmark. So that was also a really interesting finding, that we were interested in. Okay, and I'm just about out of time, so I would love to talk to you more about this project. There were a lot of things that I didn't get to mention, but in summary, we used the Tobii eyeglasses in a natural environment to find that vision is probably sufficient for learning objects in an environment this small and with this few objects. We found that longer durations of fixations led to better placement, but not for number of fixations, and that memory was better for local wall landmarks, objects that were located next to local wall landmarks. And so with that, thank you for your attention. And if you have any questions, please put them in the chat and I'll be happy to answer during the Q&A session.
Awesome. Thank you so much, Alana. Yes, as she mentioned, if you have questions, put them in the questions section. We'll get to them all at the end. Thank you so much. And now I'm excited to introduce Dr. Alexandra Hoffmann from the University of Innsbruck in Austria. She will be sharing about joint attention in a virtual team setting. Thank you so much for joining us today, Dr. Hoffmann.
Hi, everyone. I hope you can hear me properly and see me. So my research presentation today is about joint attention in a virtual team setting. And this is a highly relevant topic because, as you all might notice, we are virtually collaborating on a daily basis nowadays. Not only because of the pandemic, but also because it is easier to have connections all over the world. There has been some research on the synchronization between individuals, and this is something that improves virtual teamwork as well. So that was what I was diving into with the Tobii glasses. The glasses are, in this study, a method for hyperscanning because they enable us to simultaneously measure activity from multiple individuals in real time, not only neural activity, but in this case, like blink activity and gaze behavior. There has been studies that during social interactions, individuals align on a neural activity level, and mainly methods like EEG, fMRI, and fNIRS have been applied, but there have been some initial studies using dual eye tracking as well. So in my study, I was concerned with shared attention and shared mental models, which are which are really relevant in virtual teamwork. So there is a shared attentional focus on a third source, like a presentation or any task you might solve in your teamwork. And during this process of sharing the attentional focus, there might be a synchronization of the blink rate. And this shared attention also predicts group performance, which has been shown in earlier studies. So in my study, we applied dual mobile eye tracking as the hyperscanning method, and we quantified blink synchronization to predict group performance. And in this study, we applied a problem-solving task and visual search task in our team members. So the hypotheses were that blink synchronization would predict team performance, and that the synchronization would increase over the time course of the virtual teamwork. So yeah, we applied two pairs of Tobii glasses, but not the Neo model, but the Tobii Pro glasses too. There were three steps in the experimental procedure. So the two team members familiarized with each other for a period of 5 minutes. And then after that, they solved the problem-solving task And after that, they had the visual search task. So in the sample, there were 76 participants making 38 teams. Participants making the teams were unknown to each other before they were doing the teamwork. The mean age was 23 years, and there were 48 female and 28 male participants in the study. And the setup was like 2 notebooks attached with an external webcam, then the 2 pairs of Tobii Pro Glasses 2, and we also applied chin rests so that the head would be stable and that all the measurements that the glasses would do were pretty accurate. And between the subjects, there was a partition, so they would not see each other, but they were still in the same room to be able to control both glasses simultaneously. So in the study, there were the 2 tasks. Like the winter survival task was the problem-solving task. The subjects would solve this task via a Google Docs document. And the visual search task we executed with Open Sesame. Open Sesame was also able to control the glasses. There is a glasses controller software from Tobii, but we applied this software because therefore we could also present the visual search stimuli. For the data processing, I used a blink rate noise detection algorithm from Hirschman, and therefore we could detect all the blinks in the data. And for the synchronization of the 2 team members, we calculated the phi coefficient. The results showed that the problem-solving performance was highly correlated with, not highly, but was correlated with the synchronization of the individuals with an effect size of 0.3. That's like a medium-sized effect. And there was no significant effect for visual search performance, though. We think that this was because during the problem solving, the team members could see each other with the webcam, and during the visual search, there was no webcam view included. But that remains to be discussed. Over the time course of the virtual collaboration session, we found a significant main effect of task, and we found significant differences between the different phases of the session. So the synchronization really increased across tasks, which you could also see in the next slide. So there were some limitations in the study, and this is probably due to the mobile eye tracking. And there is a poor temporal resolution. As you heard before, the eye tracking glasses only have 50 Hertz. So with a remote eye tracker, you would probably get better data or higher resoluted data. We also tested only 2 types of tasks, and there is room for more tasks to be investigated, of course, and the sample size was rather small, so it would be feasible to test more subjects in future studies. Nonetheless, the suitability of blink synchronization is proved for predicting group performance, especially problem-solving performance in this study. And I think I showed that dual eye tracking is a suitable method for hyperscanning, and blink synchronization predicts the problem-solving performance really well, even in the virtual team setting. So if you have questions on my study, I will be happy to answer them in the Q&A at the end. And I thank you for your attention.
Awesome. Thank you so much, Dr. Hoffman. That was really interesting. I'm looking forward to hearing people's questions at the end as well. So thank you. And finally, we have Dr. Daniel Levin from Vanderbilt University in the United States, and he will be discussing eye tracking as an analytic for nursing simulation training and post-simulation review. So take it away, Dr. Levin.
All right, thanks a lot for inviting me. Real excited to give you an overview of some of the projects that we're doing. In this particular project, what we're really interested in is how eye tracking can be an online analytic that can help predict eventual learning in real world settings. Now, one of the big problems with doing that is there's really a huge gap between upfront visual attention that's reflected in eye tracking and the eventual conceptual integration that is reflected in learning. And that gap can be actually pretty surprising. So one of the big ideas that our lab focuses on is how attention is sometimes associated with representation, but not always, and how attention and representation are sometimes associated with comparison across views, but not always, and how all this stuff is sometimes associated with memory and conceptual integration, but not always. And so this gulf between upfront visual analytics and eventual learning is something that really requires a lot of careful research. And one way to solve this gulf or to address it is to get a lot of interdisciplinary people involved in your research. And so this research really involves a great team of scientists in computer science, Gautam Biswas's lab, the School of Nursing, Marianne Jesse's lab, and our lab, and also some great PhD students and lab personnel, Caleb Vitrell, Clayton Cohn, and Eduard Davalos in computer science, School of Nursing colleague, lab personnel, Eric Hall, Joanne Holt, and also Maddie Lee. And so let me tell you a little bit about the environment that we're doing our eye tracking in. What we're doing is collecting online attentional analytics in nursing simulation training. So our research takes place in Vanderbilt's nursing simulation ward, where nursing students complete exercises that involve real world equipment in this simulated hospital ward, and also mannequin patients that they interact with. They're kind of like robot patients that they interact with and they perform procedures on. And this all takes place around sort of a hospital bed that has equipment, for example, IV infusion pumps, and also monitors where the students can read vital signs and read patient charts as well. And so what we're doing is collecting a lot of multimodal data on these simulation experiences from overhead cameras, microphones, the eye trackers that we put on the students on their Tobii Glasses 3, but also computer and sensor logs as well, and combining all these data. This past spring, we did our first pass at recordings from students doing their simulation experiences. We recorded about 30 students, got about 30 minutes of usable eye movement data from each student. We had a lot of individuals and some teams. We'd like to do more research on teamwork, but we don't have quite enough control over the groups of students to get a consistent set of teams. So we're really looking at some individual data and some teamwork data. And we're doing a range of different projects with these analytics to sort of bridge the gap between upfront attention and learning. One of the projects is being run by Maddie Lee, a PhD student in cognitive psychology. And what she's looking at is how these analytics, especially gaze data, can be used for post-simulation review. This is a really big topic in nursing, helping students reflect and debrief on their experience. So what we're doing is we've been adding those attentional analytics to these debriefing experiences. And we do a really sort of intensive form of debriefing with videos that depict students' own gaze while they were doing the simulation. What we have students do is come in after their simulation, and they view their video, and it's the egocentric video with the gaze plotted on it. And then after they view the video, they segment the video. They give a response every time they think a meaningful event begins and ends. And so typically students will segment their videos, about a 15-minute video, into about maybe 40 events. And then once they've segmented those videos, they've really told us something important that helps us bridge that gap between attention and learning. And that is, what are the meaningful chunks of action that we can now collect some information on? And so we have students reflect on each event. They classify the actions that they were doing during the event. They tell us how good they felt or how high their self-efficacy was during each event. They tell us a little bit about some of their cognitive approaches, internal, external focus, tell us how successful they felt they were at teamwork during these events. And then they give some general performance ratings and reflection feedback. One really important thing I want to point out here is that this review process, and in fact, all of our research, really is founded on a careful analysis of the specific events that structure this domain, in this case, nursing simulation training. And this is in a large part due to some really great work by Caleb Vitral in engineering, who's done a really deep dive on the general kinds of events that happen during simulation training. So for example, information gathering, intervention, support, and assessment, and also more specific events that occur during this training, ranging from sort of preparing an infusion pump to reading orders to looking at vital signs. So we've got coarse grain events, the big events, but we're also looking at the fine grain events too. And Caleb did this work in collaboration with our collaborators in nursing. And so you can get a lot of really interesting data out of these segmentations that the students do after the events. One nice thing about it is that all by itself, the segmentations and then the classifications that the students do can be useful information, for example, for nursing instructors. One thing Maddie did was she analyzed the segmentations for a student who gave themselves a very high performance rating, felt very confident about themselves, and also another student who gave themselves a very low performance rating. And a whole bunch of things jump out of these segment classifications. So if you look at the top, we have the abstract action classifications. And we see one big thing that happened with the student who had low confidence is that they weren't doing anything for a while. They had a no activity classification. And another interesting thing that happened with this low confidence student is they actually didn't perceive very many events. And so these kinds of results can be used to sort of show nursing instructors what's going wrong in the simulations in sort of a pretty compelling way. Another really nice thing about these segmentations and classifications is that the students themselves are giving us some really nice data, not only to understand eye tracking, but also to do things like machine learning, to train classifiers that can automatically start detecting events because they're naturally telling us what the event is, when it begins, when it ends, and what's going on during that event. So I've done a bunch of analyses on these data. One analysis that we recently did just took a look at properties of each of these events that the students have identified and see how they predict important outcomes for each event that our nursing colleagues really care a lot about. One of them really is that sense of self-efficacy. This is a really big sort of a value of simulation training. So for each event, like I said, students rated their self-efficacy, and we've been doing some multi-level analyses looking at the degree to which self-efficacy is more or less likely during different sorts of events. So those task variables reflect the tasks that were going on during the event, for example, communication, actual actions, things the nurses were doing, and then reading and writing, reading charts and writing things. And one of the things you can see right away is that students have a much higher sense of self-efficacy when they're communicating and when they're engaging in actions. We're also looking at some cognitive variables, the degree to which they feel like they're internally focused or externally focused. There are some occasional relationships between that focus and self-efficacy. Another thing we're looking at is the degree to which transitioning to a new task is associated with self-efficacy. And finally, we have been looking at some gaze statistics, for example, saccade amplitude and fixation duration. And as probably many of you know, there's a lot of circumstances where you see no relationship between the basic gaze statistics, and there are some circumstances where you will see those relationships. One of the things we're really trying to do here is not just calculate gaze statistics across an entire timeline, but really sort of get gaze statistics as articulated across specific events in that timeline. Another variable we've been taking a look at is teamwork. This is also really important to our nursing colleagues. And so this reflects for each event how, to what degree the student felt they were working individually or as a team during the event. And one of the really interesting things you see there, obviously, is communication highly predicts the amount of teamwork, but you do see some prediction for the gaze analytics as well. For example, segments where students rated high teamwork were predicted by relatively large saccade amplitudes. Fixation duration did not predict teamwork. One other thing we're really trying to think about is getting beyond these basic statistical relationships between gaze statistics and outcomes to try to figure out if there are repeatable sub-events that we can detect that might moderate the relationship between things like fixation duration and outcome. So for example, if you analyze what's going on during these events, you'll see situations where, for example, language is really guiding gaze. And so this is an event where the robot patient, as run by the instructor, said, "Is the blonde girl at the foot of the bed feeling better?" And the answer from the nurse at the foot of the bed was yes. And so you can see that some of the saccades and head movements really were invoked by the language that was produced by the patient. So is the blonde, foot of the bed, that the hair color and the location directly guided saccades and then fixations. And then there was a funny little moment where there are actually two blonde nurses at the foot of the bed. And so the nurse wearing her eye tracker moved her head and saccaded over to the first blonde nurse and realized that wasn't the one that was being referred to, and then moved her head and saccaded over to the second blonde nurse. And so that's a situation where processing one object has led you to shift your gaze to another object. And so what we're trying to do is identify the degree to which there's repeatable sub-events in these timelines. And I'm going to skip over the big idea here and sort of figure out, number one, do these repeatable time events happen relatively frequently? And they seem to. We can detect a lot of them. And they seem to reflect a good proportion of the timeline. Our first pass, we were able to classify about maybe 20% of the timeline with these potentially repeatable sub-events, although stay tuned for the research on this. We're not quite there yet because we have not succeeded in training undergraduates to code these reliably. So more news on that as we go on. Another thing we're doing is, as you might expect, trying to figure out if we can process gaze or do research on gaze by looking at the degree to which gaze falls in specific regions of interest. And of course, as many of you I'm sure know, it would be really great to be able to process and identify those regions of interest without having to do it manually. And so this is what Eduardo Davalos has been working on, processing that egocentric video using deep neural net learning, the YOLO model, to try to identify regions of interest. But one of the things he really wants to do is, in addition to identifying those regions of interest, trying to figure out the degree to which you can detect different events and transitions between events by taking a scan path graph approach to figure out if there's repeatable visits to locations and transitions between locations. So what Eduardo has done is he's taken some of our videos, done some object recognition, and then classified area of interest sequences based on the objects that the students are looking at. And what we're looking at here is 3 different nurses who have all worn the glasses through the same sequence of events. And so he's found some ways in which, so the first step is to detect the ROIs, and then you try to group them using a scan path graph approach. And how that works is you go through the timeline with a series of 15-second windows. And within each window, you characterize, number one, the amount of time that gaze falls within each region of interest, and then also the number of transitions between those regions of interest. And you represent those 2 properties in these information graphs that you can see on the right there. And so the size of the dot in the information in the graph reflects the amount of time in the ROI, and then the size of the arrow reflects the proportion or the number of transitions between those ROIs. And what you can do once you have a series of graphs reflecting each sort of segment of the video is you can try to figure out if there are clusters of graphs by basically comparing the similarity between all of the graphs and figuring out how many clusters there are. When Eduardo did this, it's through using k-means clustering. He found about 5 clusters. And some of those clusters really seem to reflect the initial diagnostic things that the nurses were doing. And you can see one of the things in this diagnostic cluster that's going on here is there's a lot of looking at the monitor. And other forms of diagnosis that he classified as general diagnosis, more looking at, in this case, the person and the bed they're in. And so using this approach, you can classify sets of AOIs by a higher level cluster and try to figure out if that classification really is associated with meaningful events. And as a first pass, and we've got a lot more work to do here, Eduardo has found that, yeah, there are some ways in which these clusters seem to reflect meaningful events. For example, those diagnostic events seem to occur at the beginning of the timeline, as does information gathering. So you can see the code here on these clusters for these participants reflects information gathering, and that's that reddish brown sort of set of clusters, and then diagnostic information gathering, which is the more, what is that, mauve, I guess, clusters. And then some other clustering goes on throughout the timelines. And one thing we'd really like to do is then articulate or compare these events as detected by the scan path graphs and the events as sort of classified by our actual participants to see how effectively they correspond. And so we're doing these projects and a bunch of other projects, looking at developing online multimodal analytics integration, looking at trying to link gaze patterns and specific events. We're going to be doing some language processing that as an aid for event identification and some interaction analysis of behavior sequences. So overall, we're really trying to sort of take those analytics and bridge the gap between attention and learning. And one thing I want to end with is we've gotten some support from the Live Center here at Vanderbilt, which is a center that we're just starting up to do research in learning and technology, and also to get that research out there to help commercialize it. And for those of you on the call, if you have any folks who might want to do some work in this area, get in contact with me. We're hiring, especially if you have an undergrad with a BA coming out. We're looking right now for a research assistant. Have them get in contact with me. And thanks for listening.
Awesome. Thank you so much, Dan. And really great to hear about your partnership and job opportunities. So please feel free to reach out to Dr. Levin. At this point, we're going to start our roundtable discussion. I'm going to invite all the presenters to come back. So please, for everyone who's in the audience, are we seeing my screen again? Let me see. Are we seeing the QR codes up on the screen yet? Okay, great. Okay, so again, all the attendees, please keep putting the questions in the chat box. For the QR codes on the screen, if you are not an existing customer or don't have eye tracking in your lab, but you're interested in it, you can reach out to us and we'd be happy to chat with you about your research, recommend the right product for you, and provide budget. So the first QR code on the left is the contact form to reach out to us. The QR code on the right is to talk to one of our team from our funding support services. This is the team that I manage in the United States. We have these funding support services designed to assist researchers who are looking to integrate eye tracking into their methodology, but they don't have the money yet and they're working on grant applications. So this is a free service that we provide. We can help by giving letters of support, individualized budget justification for our Tobii solutions, feedback on the eye tracking components of your funding proposal, and so much more. So we're here again to provide this free consultation, and feel free to reach out to us with any questions. For those of you on the call who are existing customers, you can please make use of Tobii Academy and Tobii Connect. You can find those both at connect.tobii.com or academy.tobii.com. Academy is our e-learning platform. So it's self-paced training for you, students, and any collaborators that you have. And then the Tobii Connect portal has a ton of white papers, questions and answers, and that's where you can also access the support team. And then when we send out the recording link for this, we also have a white paper and a blog post about cognitive psychology that we're going to include in that follow-up email. So feel free to check that out as well. Before we get started with the Q&A, and as everybody keeps answering their questions, I have a couple of questions for the audience. The first one is, have you used eye tracking in your own research? So the first answer is yes, I'm currently using eye tracking. Yes, I've used it before, but I'm not using it right now. Or no, I've not started using eye tracking yet. So for all of the people in the audience, if you would tell us, are you currently using eye tracking, interested in it, or haven't started yet? Love to hear where everybody is in their familiarity with it. Looks like so far we have a lot of people who are currently using eye tracking, which is really cool. That you're using eye tracking and then interested still in what other people are doing for their research. So I hope this has been inspirational for you so far. Okay, I'm going to close this now. One more second to get people's results in, and then I'll share the results so everybody can see. We had about half who are currently using eye tracking, about a quarter who've used it before but not currently, and then about 20% who've not used eye tracking yet. So we hope that you will some point soon. We have one more question about fields of research. So generally, are you from a cognitive psychology area, some other area of psychology? Are you from neuroscience or some other fields? And if you are from another field, we'd love to hear in the chat box what your field of research is, so we can point you to some other resources for you specifically. Awesome. Looks like we have a lot of people in the cognitive psychology area as well. Okay, give it like 3 more seconds. Okay, great. Thank you all so much. Let's look at those results. So a good chunk in cognitive psychology, some other psychology areas in neuroscience, and then almost half are from some other fields. So I hope to hear what kind of fields you're from. If you feel free to share, we'd love to hear. Awesome, thank you so much. So I have questions now for the panelists. Let me take the poll results off. Okay, so just a general question for everyone of my own interest. What's the most compelling part about using eye tracking as a research tool? I'm sure many of you have done studies that didn't include eye tracking at all, or maybe you added eye tracking in later in your careers. So what is eye tracking added to your research that you couldn't get from other methods? So maybe we'll just start with Alana. I'd love to hear a little bit more. You mentioned in your presentation about considering VR, so I'd love to hear a little bit more about this from you.
Yes, my lab actually does a lot of VR, and they have one of those omnidirectional treadmills that I showed a picture of. And I do want to use those technologies, but when you're in VR, everything looks so different from the real world. And there are measures of immersiveness that you can have for VR. So it's not, I'm not saying that VR can't tell us a lot about the real world. It can, but you can't get closer to the real world than the real world. So it was kind of a new venture for my lab to get eye tracking glasses and to try more realistic visual stimuli. And so that added a lot to my lab and kind of opened up a new track that I could then explore.
Awesome, that's really great to hear. How about you, Alexandra?
I think I started using eye tracking in my PhD with majorly depressed patients, and it was a screen-based eye tracker from iLink. And then after that, I got into like the cognitive psychology again, and I started using the Tobii eye tracker, the screen-based one. But we had the glasses also, and my supervisor said, "Yeah, why not use the glasses?" And then I found that investigating social interactions and face-to-face interactions was really even more interesting than all the screen-based things you could do. So yeah, the next project I plan on doing is with patients from the eating disorders psychiatry station. So I hope to gain really important results for social interactions and dynamics, and even in disordered individuals.
Awesome. And finally, how about you, Dan, to that question about, what is eye tracking added to your work?
Well, I think on some fundamental level, one of the things I like about it is you don't have to wait till the end of the event to find out what people were thinking when they experienced an event. So they could watch a movie, they could experience an event. And our research on visual attention often tries to make inferences about what was going on while they were experiencing the event, but we only have responses from the end of the event. And so eye tracking is one of those ways where you can really sort of go into the event and find out what people were thinking, or get some at least signs about what they were thinking without interfering with their experience of the event. Of course, you can have them segment events, and we do that a lot, but that really is a form of processing you're adding to the event. And so if you can have a less intrusive means of understanding what's going on, I think that's something that really offers some interesting opportunities.
That's great. That's very insightful. Just the immediacy of those results. Okay, I got a couple other questions for everybody. So Alana, excuse me, I'm kind of recovering from a cold. I'm almost better. Alana, you mentioned in your study, or in your presentation, about some technological advances, especially in eye tracking, that have allowed for you to do the kind of mobile wearable research, like that chin rests aren't necessarily required. Excuse me, at least for our eye trackers. So I guess sort of dreaming in the future, what kinds of eye tracking capabilities would you want to see for the future, for take your work forward even more?
It's a great question. I think one of the things I would like to see is having it adapt better to natural light, because I would love to take these eye trackers outside, and the only thing stopping me is I need to find either a big enough hat or a big enough visor to really cover the sun that comes out in Arizona pretty much all the time. Other than that, some of the software, I'd love to see more capabilities analyzing it. I think Dr. Levin mentioned scan paths, and I'd really love a tool that made those a little bit more accessible. And I don't have an eye tracking background except for what I've done in my PhD, so it's been a little bit difficult trying to get up to speed without somebody who really knows the material to help me. So I've relied a lot on the Tobii software because it is pretty user-friendly. But I would love a scan path tool, and also I would love to have a list of fixations and durations and just throughout my experiment, instead of just an average at the end.
Awesome. So Alexandra, you mentioned that the participants in your study were strangers. So I was curious, what kind of blank sync results would you expect from participants who were close friends or relatives? And sort of what was the rationale of picking people who were unfamiliar to each other?
So I think that was based on other studies measuring synchronization between individuals. So if you are familiar with each other, you synchronize better and faster with each other because you know each other better. And yeah, that was just because other studies did the same methodology.
Okay.
It seemed to be a common standard. Yeah.
Okay, interesting. And Dan, I wanted to talk about your partnerships, because I think that eye tracking is an awesome tool for collaboration, and we started to see more research at universities partnering with people from other departments. So I was really happy to hear about that with your partnerships with computer science and nursing. So can you tell us more about how those partnerships came to be?
Yeah, so we, I guess it got its start just when I approached our nursing colleagues because we were doing instructional videos and just wanted some good contents for our instructional videos, and we were hoping we could find some procedures that they teach their students that we could really sort of have some nice videos that show actions and have some clear learning goals that are easily testable. And so we started interacting, making some of those videos, and then we realized, the nursing folks really showed me, look, we have this huge sim ward, and I didn't even know about it. And they gave me this tour, and it kind of blew my mind that this whole hospital ward is up there, and that they have their students really walk through and learn a lot about doing these simulations. And so we realized it'd be really interesting to track visual attention in these settings. Our first instinct was to do research on safety. That's closely associated with my previous work on sort of change blindness, failures to notice salient things in your environment. Because one of the big things that happens in these settings is that individuals fail to notice safety critical things, like, for example, the bed rails being up. And so we thought, wouldn't that be interesting to really do some visual attention work to see sort of if we can predict failures to detect safety issues. And we started working on that. We kind of realized it was really hard to get a lot of data on these individual moments where you might fail to see the bed rails being out or whatever it was. And so we really started rethinking what we were doing to really sort of get more data and do more analyses on all the stuff that was happening in these simulations. And so that's why these post-simulation reviews seem to be useful, but also there's a lot of great engineering issues associated. Gautam Biswas's lab is really interested in extracting the meaning from these analytics. There's just a lot of opportunities to do that in really interesting ways as well.
Awesome, that's really cool. I hope that is inspirational to other people who are considering partnering with people in other departments or colleges, especially with eye tracking. Great, so another question for Alana. Have you tried to use a pharmacological challenge and measure the memory impairment on eye tracking? Guessing some sort of medication intervention. Have you considered that?
I haven't yet. The main reason being I'm more focused on not clinical populations, just kind of the normative young adult healthy populations to just get a baseline first. My lab does work with some clinical populations, and I am going to do this study in older adults soon who might have a little bit more of a memory decline, not impairment. They're still healthy aging population. But I think I might finish my PhD before I get to do that. So possible post-doc work potentially in the future.
Yeah, definitely. Awesome. So now a question for you, Dan. Did you try to apply the sub-events analysis to psychiatric population with visual integration impairments, for example, those in schizophrenia? So I guess another sort of clinical question for you.
Yeah. I mean, that's a really interesting idea. One of the ideas that sort of came to mind to us before schizophrenia, which is definitely relevant, is individuals with autism spectrum disorders, especially if you start looking at the kinds of things that occur just after you follow gaze cognitively. There's some research suggesting that individuals with ASD will follow gaze, but then there's the inferences that happen after you sort of follow gaze that might be really interesting to see if there's some differences there in fixation patterns that might be related to those sub-events. So we've thought a little bit about that, but it's a great idea. We're trying to get the sub-events to work, though. We have not gotten them to work. So a work in progress, but we're really hopeful that we can have that as a mediating factor. I know Alana mentioned Ballard and Hejo's work, and that was really inspirational for us as well to think about these sub-events, because that's really one of the big things that they've been talking about for quite some time as well.
Yeah. Cool. Another question for you, Dan, about the nursing study. Saccades happen very fast, so why did the segmentation happen in 15 seconds, not 10, or what's the rationale behind the 15-second time series?
Yeah, I mean, I think that it was determined to a degree empirically. Eduardo tried several different durations for those time windows and found if the duration is too long, the event just never changes. And if it's too short, you don't have enough saccades, and they're not. He isn't really doing event perception. It's not fixations. It's just time in the ROI. But you just don't have enough saccades and visits to ROIs to get a stable estimate of how this graph would look. So if your time window's too slow, you just don't really have enough data.
Gotcha. Okay, and then if anybody else has other questions, feel free to add them in. We have one more. I think this might also be for Dan because it's about learning, but which eye tracking metrics are useful indices to demonstrate improved learning?
Right, so there's $64 question. We're trying a lot of things. Traditionally, a lot of those metrics have looked at intersubject correlations. Has been a very powerful way of assessing the degree to which someone's doing a good job learning because they've looked at other things that people look at when they look at it. And so that's one way to get directly at learning, especially if you've got teams. But more generally, we're hoping that some of the gaze statistics, especially if we can have those sub-events as moderators, might predict learning. And I'll be clear, I think it's an iffy venture. It may work, it may not work. So that's one of the reasons why we're also interested in working on these post-simulation reviews and assessing the degree to which, for example, gaze can predict other important things like that self-efficacy. I mean, that's a really important part of these nursing simulations as well. And if we can find some more of those links, not only between learning, but also with other important variables, we're hopeful that we can have a way of detecting online when things are going well in the simulations and when they may not be going so well.
Great. How about Alana or Alexandra, if you have any follow-up to that as well? Or any insights into...
That's a great answer.
Okay. Awesome. Just wanted to read off a couple of the fields that people mentioned in the chat. We have people from cognitive and information sciences, linguistics, geovisualization, and navigation, which would be really, I'm sure they're really interested in your study, Alana, particularly. Human-computer interaction usability, and vehicle engineering. So some really cool other fields outside of cognitive psychology. Any other final thoughts from any of the panelists or final questions if any of you have anything you want to share? I think we'll be wrapping up soon, so the floor is yours.
Yeah, I think one of the things that I've been thinking about a little bit is that as this sort of head-mounted or wearable eye tracking has developed, you see these rather small incremental improvements in the wearability of these things, the accuracy, the quality of the video is just something we're obsessed with too. It tends to be low contrast video, or high contrast video, right? So it gets blown out a lot, we lose pixels. And so each of those little increments really kind of seem like the thing that's going to matter the most at this point in getting that stuff to really be useful and work. That's my sense anyway. So the little improvements reflected in the Tobii Glasses 3, they're kind of important for getting this stuff to work out well.
That's really great to hear, and hopefully whenever the next generation comes out, it'll be even better and make your research lives even easier. So thank you all so much for being here today. Thank you to all of our attendees as well. Again, this is recorded, the link will be sent out soon, and we hope to see you at the next webinar that we'll be hosting. Have a great day, everyone.
Bye, everybody.
Bye.
Bye.
Thanks for having us.
Thanks.
Thank you.